All right, so in the last lecture, I want to focus the attention on, well, kind of the very last step of the computational analysis pipeline for single-cell RNA-seq. So this, again, is this overall pipeline that we have gone through. Once we have the gene cell count matrix, we've performed clustering analysis to get cell clusters. Then often the next step is biological interpretation, like the clusters we get. How are they different from each other? What kind of biology each one of them represent, right? So in the literature, I think the most popular approach for doing this is actually by manual inspection of cell type specific marker genes. So for example, one thing we can do is, let's say we have the data already clustered, and then we can perform differential expression analysis for each cluster against everybody else. So we can do cluster 0 versus combination of all the other clusters, cluster 1 against combination of all the other clusters. So we do these DE differential expression analysis, and we'll get top markers that are highly expressed in cluster 0 compared to the rest, and then marker genes for cluster 1, et cetera, et cetera. And just by looking at the names of these genes combined with prior biology knowledge, it is possible for us to come up with suggestions or ideas of what each of these cluster, what kind of biology they represent. So this is one way to do this manually. Alternatively, another way to do this is we can, well, take the UMAP visualization and then try to color code it according to the expression level of a particular gene, which is a marker gene that we know can help us to identify certain cell type. For example, MS4A1 is a marker gene for B cells, so we know that already. And according to that prior knowledge, what we do is we can color code this particular UMAP, right, with expression of MS4A1, and we see this island showing high expression and no expression anywhere else except just some scattered noise. So then this gives us a strong indication that this cluster of cells is B cells, right? We can do that using other markers for monocytes, for T cells, for NK cells, etc. And that can help us to identify cell types, okay? Or, I mean, cell type annotations for these clusters. Now, this process, it's time consuming and it's also labor intensive. Imagine if there are many data sets that needs to be annotated, and going through even these relatively straightforward process manually can take a long time, and hence, you know, there is desire to get this whole process automated. Okay, so one idea to develop automated algorithm to perform cell type annotation is to, in some sense, mimic the biologist's thinking process for annotation, right? So for example, we can take a database of prior knowledge that we know B cells have these known marker genes, T cells have these known marker genes, et cetera, et cetera. So if we can come up with such a database, actually there exists such database in the literature. One is called Cell Marker, and also in the cell ontology database, there also exists a lot of information on which cells are defined by what kind of marker genes. Now, once we have this database as our prior knowledge, what we can do is we can try to say, examine a new single-cell RNA-seq data set and ask for each cell here, or maybe each cell cluster, what kind of genes do they express, and then use our prior knowledge to guide the annotation, but in an automated way. So one thing that makes this approach difficult to automate is that, well, the number of established cell type specific genes or marker genes are often very small for any given cell type. Okay, and because that number is small, and because single-cell RNA-seq data has high sparsity, it is possible that, you know, using just the small number of known marker genes is not sufficient to accurately annotate you know, various cell types. And also, very often, cell types can be defined by combinations of multiple marker genes. for example, gene A positive, gene B positive, and gene C negative. Maybe that combination is what defines a certain cell type. This is especially true when you go to very detailed immune cell types. For example, B cell subtypes, T cell subtype, those naive, memory, effector, regulatory subtypes. Their definitions are not always based on positive markers they express, but very often their definition contain these negative markers. But then, we have to really develop algorithms to, well, kind of recognize both directions in order to fully utilize existing knowledge, right? So,
there's a clarification question in the chat. Do you mind?
Okay, let's see where's.
Maybe it refers to
on scale of the plots. Oh, okay, the unit for slide 3. If we go back here, I guess, right? So these units, I believe they are gene expression levels after log normalization. So, so you can think of these as 0 means 0 count, 1 means a count of 1, and 2 means a count of 3, because 2 raised to the second power, right? And then 3 means a count of 7. Right? And in this color bar, the darker color you see, that means higher gene expression level. Okay, so I hope that answers this question. All right, so regarding strategies for developing automated cell type annotation algorithms based on such prior knowledge. So one idea is that we can take individual cells gene expression profile and then compute something called single cell gene set enrichment score against these known gene sets, and then use the enrichment score to make predictions for cell type labels. This is actually an approach that's taken by these two algorithms. One is called SCSA, and the other is called SC-catch. So for both algorithms, the way they operate is that they first cluster the data so that given this new data set, they derive, say, these 100 cells belong to the same cluster and these maybe 500 cells belong to another cluster. And then what they do next is they perform differential expression of one cluster against all other clusters combined to identify genes that are highly expressed and also lowly expressed, relatively highly or lowly expressed compared to the rest of the data. And then they use these DE genes to compare against no marker genes and then see whether there's alignment. If the answer is yes, if the DE genes for a certain cluster happen to overlap a lot with, for example, B cell specific genes, then that cluster is annotated as B cells. Okay, so in fact, at this moment, I have a student in my lab who's working on a comparison between these two algorithms and, well, Garnet and Azimuth, which is a Seurat-based algorithm. And it turns out that these two algorithms, based on sort of cluster data, they are actually not performing really well. I mean, not well, it's actually performing very poorly. And we actually have some hypotheses of why that's the case. But we feel that their general approach, direction of cluster data first and then followed by annotation, I think is actually a good direction to go. It's just their implementation, well, have room for improvements. Okay, but anyway, so a third approach for utilizing prior knowledge is to use these prior knowledge to manually define a decision tree and then use the decision tree to classify cells into known cell types. And this is the approach taken by a group actually based in UW, and the method they developed, they named it as Garnet. So this approach to me is actually very nice and very intuitively aligned with how biologists are operating. Because, for example, if you think about flow cytometry data analysis, right? So what people usually do is they use known markers for example, CE3, CE14, CE19, those known markers for major lineages, and then use those markers to draw gates or make decisions of whether cells belong to, say, the lymphoid lineage or the myeloid lineage or the B or T lineage. And then after making that first level of that high level decision of which lineage cells belong to, and then they use other markers to try to further separate cells into subtypes. So the hierarchical decision tree approach that Garnet takes pretty much follows the flow cytometry analysis. But of course, I mean, the difficulty for using this algorithm is that the users will need to define the decision tree and the kind of the gating hierarchy. So I think the people who develop Garnet, they actually already constructed a few decision trees themselves for the immune systems, and I think for also various neuronal cell types. So that this tool, well, if you are interested in, say, if your data is generated from PBMC or bone marrow, is about immune cells, then you can directly use their tools. But if your data is about a, say, specific biological contexts that's different from what the Garnet authors provide, then you will have to generate a decision tree using your prior knowledge, and then you can use this tool. Okay, but these are, again, some example algorithms and the general strategies of how to design a automated algorithm that can help you to annotate a new data set's cell type label using prior knowledge of cell type marker genes. Okay, so another approach that's, I think, more popular in the literature is a fully supervised machine learning framework, where we say that we have a reference data set, a reference single-cell RNA-seq data set that's previously generated and is previously clustered and well annotated so that we know what is the class label associated to each cell. And then now we can use supervised machine learning algorithms such as SVM, random forest, and or neural networks. Right? Use these approaches to learn relationship between the data and the labels. Once the relationship is learned in the form of some kind of function, then we feed this new data into the function, and the function will help us to predict the class labels for every cell in this new data set. Okay, so this is intuitively very straightforward in terms of the machine learning framework, but implementation-wise, this is actually not very easy to do, because it is possible that this reference data set and the new data sets, they're generated by different single-cell technologies, so that there may exist a very strong batch effect, or it's possible that the reference and the new data set have some mismatch in terms of the biology. Maybe they are coming from about similar cell types, but coming from different tissue sources. For example, colon versus ileum, right? Or maybe the reference and the new data sets, they are again about the same biology, but then under different disease condition. Maybe reference is healthy and the new data is a disease sample. Okay, and well, for both of these reasons, I mean, their consequence is that there will be some batch effect in between the reference data and the new data, so that the relationship we learned down here, when we try to apply that relationship to new data, it's not directly applicable. So one example is actually in computer vision. So well, you guys probably know that, heard that deep learning has been sort of revolutionizing the computer science field. And now we can easily build classifiers to classify images into different types of objects, like cats and dogs, cars, planes, boats, etc. Right? And it turns out that, you know, if you have, let's say, some previous training data, those are all, say, gray images without any color, right? And then you have millions of such images of different classes. You'll learn a machine learning model to predict these images and give them class labels, the examples I just mentioned. And now if your new data is colored images, right, with RGB channel, and suddenly and we try to feed feed the new data into the classifier learned from the deep learning classifier learned from the gray images, things will actually perform very, very poorly. Okay, and it turns out that even if we are dealing with, say, data images generated by two different types of cameras, there could be, well, that little bit of mismatch between the two types of cameras, right? The technology, the CCD behind it, can cause some deterioration of classification or prediction performance on the new data. Now, in single-cell RNA-seq, as we discussed actually yesterday, the batch effect between two different data sets, even generated by the same technology, but from different labs, could be very large. So very often what we see is that off-the-shelf machine learning algorithms applied to the training data, to the reference data, cannot really generalize very well to the new data set. And also, there is another, I think, very big issue, which is on the scope of the reference and the new data sets, and also the level of annotation. So for example, our reference data set could have only these high-level annotations of major lineages and major cell types. Then this would be a relatively easy reference data set that we can try to use to predict new data. But then our predictions can only be these major lineages, right? But if our reference data is annotated in a very, very detailed way, and we have, for example, 50 different immune subtypes annotated here, then the differences between these annotated cell types could be very subtle, and then learning the relationship between the data and these very detailed annotations could be difficult. Okay, so now when facing these difficulty, general strategy that people use, well, there are two of them. So one is that we can perform data integration and batch correction and try to say, okay, so there's strong batch effect in between these two so that they're different, but let's try to apply data integration algorithms, existing ones, so that we can merge these two data sets in a way that shared cell types are overlapping and then unique cell types are, well, kind of well separated. So if we can merge these two data sets in such a meaningful way, then and remove batch effects, then classification problem will become very easy. It's just going to be a nearest neighbor algorithm, where we after the merge for each cell in the in this new data set, we ask who are its nearest neighbors in the reference data sets and just use those labels to annotate this new cell, right? So the first strategy, this the performance of it, kind of 100% relies on the data integration algorithm's ability to remove batch effect, right? And this second approach is to identify robust similarity metrics that are invariant to batch effect. Okay, so that when we are calculating that kind of similarity metric in between cells here and also in between cells across these two data sets, these maybe correlations or whatever similarity metric, they are not really affected by batch effect at all. So if we can find such invariant similarity metric, then we can use these metric to help us to find kind of connections in between these two data sets, which cells here correspond to which cells here. Okay, and now, well, actually, if you think about these two approaches, right, integration to remove batch effect or identifying invariant similarity metrics that are not afraid of batch effects. So these two are, well, I think fundamentally sort of the same mindset, because if we can find invariant similarity metrics, then we can use those invariant similarity metrics to remove batch effect, and actually vice versa. So, so to me, in the literature, this the approach that aims to use a reference data set to learn from an annotated reference data set to make cell type annotations for new data set, it's a data integration batch removal problem. Okay, now this is a problem that I'm very much interested in, and my lab is developing, well, at this moment, two different algorithmic ideas towards solving this cell type annotation problem. And kind of following the general suggestion I made in an earlier lecture, before diving into this specific topic, we first did a literature search for benchmarking papers that compare existing cell type annotation algorithms. And it turns out that there exist 4 benchmarking papers on this topic already, and each of them compared more than a dozen algorithms. Of course, there are overlaps, but it does exist a good number of existing algorithms for cell type for automated cell type mapping. Now, so then one thing we did was we went through these existing algorithms and did a quick summary of each of them. The text, of course, is too small for you to read. I also don't really intend you guys to read it on the fly, but afterwards you can kind of go through this table and get some idea of what the general strategy taken by each of these individual algorithms. Now, after kind of going through all these existing algorithms, one thing we notice is that, well, majority of the existing algorithms, they are, well, a good number of them, they're data-driven. So they belong to kind of the second type I just mentioned, using a reference data set, learn from a reference data set to predict new data set. But there does exist a few algorithms that are knowledge-driven. So these algorithms, they use known cell type specific marker genes to form some kind of database or decision tree to make automated cell type annotation for new data sets. Okay, so and another feature that I felt worth summarizing is whether or not these algorithms have ability to label unknown cell types, because when you're trying to say annotate cell types in a new data set that you just generated, there always can be a possibility that your new data set contains some cell types that do not exist in any of the previously known kind of cell types or knowledge, right? So then it actually would be an important feature for any cell type annotation algorithm to be able to label cells as unknown, okay? And so you see many yeses here and also many no's, but there only is one algorithm that I really highlighted that I felt is special. That's because, for example, for the Surratt and Azimuth algorithm, it is able to label cells as unknown, but it's doing that in a sort of a passive and indirect way. By that, I mean, for example, if we have a reference data set that has 10 known cell types already, okay, and then when Surratt tries to use this reference to annotate new cells, a new data set, then for each new cell, Surratt will try to predict what is its probability belonging to cell type number 1, what is its probability belonging to cell type number 2. So Surratt will predict 10 probabilities for this new cell belong to each of these 10 cell types, and these 10 probability they sum up to 1, right? And what Surratt says is if the highest probability among the 10 is very high, it's like 0.99, then the algorithm will confidently label this cell. But if the highest probability among the 10 is only a 0.2, right? So then Surratt will say, this cell, I cannot annotate it. I cannot confidently annotate it as one of the 10 known cell types. So I'm just gonna label it unknown, right? So this is what I mean by a passive way of labeling unknown, because the unknowns are labeled, or novel cell types, right, are labeled only in the cases where the algorithm failed to confidently annotate it as any of the known cell types. So I think this logic is fine, but I think a more active way of annotating an unknown is that, for example, if I have a reference data set of 10 cell types, then when I try to annotate a new cell, I actually want to directly produce 11 probabilities. What is the probability of this new cell belonging to each of the 10 cell types, plus the probability of this new cell really represents something novel? And I want these 11 probability to sum up to 1. So if an algorithm is designed this way, then I would call that algorithm to be able to explicitly and sort of directly label unknown. And it turns out that among all the existing algorithms listed here, only one algorithm is able to label unknown novel cell types in a direct way. Okay, and another feature that I thought worth summarizing is that majority of the existing algorithms, they are what I call cell-based. So they make predictions for they make cell type label predictions for each cell separately. Now, this certainly will enable these algorithms to have good resolution, while at the same time, the data, the gene expression profile associated to each cell is highly sparse and noisy, and therefore, prediction based on individual cells may potentially be less accurate than predictions based on clusters. Okay, so these three algorithms, they are special because they cluster new data first, and then they make predictions not for individual cells, but for each cluster. So the two algorithms I just mentioned, SC-Catch and SCSC, they're designed this way. And conceptually, I would say that they should have higher and more accurate cell type annotation performance. But when we tested them, we feel that their performance is far from satisfactory. And we are actually currently developing algorithms to kind of follow their general idea. We cluster the data first, and we try to annotate clusters instead of annotating individual cells. Okay, and another column here, another sort of feature that I think is worth mentioning is whether or not these algorithms can utilize multiple reference data sets. Because while single-cell RNA-seq has been out there for, 7 years already, and people are routinely generating more and more and more single-cell RNA-seq data and making them available, very often also along with the author's clustering results and annotations, right? So now in the literature, actually, if we want to find, say, an annotated PBMC single-cell RNA-seq data set for reference, there probably exists more than a dozen. Right? And these data are generated by different technologies, and they're analyzed by different people, and they're of course labeled by different people, right? And now, most of these algorithms, they can only work with one reference data set. But then among all the possible reference data sets out there, which one is the best we should choose, right? And also these individual reference data sets coming from individual groups, who's to say that their analysis is 100% bulletproof? Right? So maybe any existing data set we pick as reference, there might be small errors in there in terms of their clustering results and annotations. So ideally, a good cell type annotation algorithm based on, say, existing reference data should be able to work with multiple reference data sets. Okay, and if the algorithm is utilizing many reference data sets, and hopefully, even though each reference may contain some error, but their consensus could be really a comprehensive and accurate atlas that we use as reference to annotate new data. Now, among the algorithms listed here, I think only one of them is really explicitly designed to utilize multiple reference data sets in a sort of in an integrated manner. Okay? Algorithm development is that we want to try to develop an algorithm that is able to explicitly label unknown, and then try to make based on cell cluster multiple reference data sets. Then we can say that this new algorithm at least have some conceptual advantage, right? So that makes sense. Okay, so and our idea is that we want to kind of build a algorithm that's combining knowledge-based approach and data-driven approach. So our idea is that, given a reference data set that's already annotated, okay, what we can do is that we can perform differential expression analysis between each pair of cell types in the reference data, so that we know which genes are differentially expressed between B cells and CD4 T, which genes are differentially expressed between B and CD8 T cells, et cetera, right? And these collectively, these represent the knowledge that we can extract from a previously annotated data set. Okay, so this way, our algorithm based on such knowledge, it hopefully will be more extensive than the cell type marker genes that let's see, the cell type marker genes that were curated in the previous literature, right? But anyway, so these are the knowledge we can derive from one reference data set. And if we have multiple reference data sets, we can derive more such knowledge. And then for the new data set that we want to annotate, we propose to cluster them first. So that's the same as some of those algorithms on the previous slide. Okay, so we have these cluster labels. Let's just call them 1, 2, 3, 4, 5. And then what we can do is that we can hypothesize, or we can make a guess of how these clusters should be annotated. So for example, our guess could be that cluster 1 and 3 are both B cells, cluster 2 is actually a novel cell type, et cetera. So we can make these guesses. And after we make this candidate annotation, which may be correct, may be incorrect, but then what we can do is that we can take the new data and combine with this candidate annotation to derive the same knowledge. So for example, since our annotation says cluster 1 and 3 are B cells and cluster 4 are CD4 T cells, so if we want to derive knowledge about DE genes between B and CD4 T based on the new data, we should be looking at differentially expressed genes between cluster 1 and 3 combined versus cluster 4, right? And same goes for kind of the logic of this table. And now, if, okay, our idea is if this candidate annotation is really correct, if cluster 1 and 3 are really B cells and cluster 4 is really T cells, then the DE genes generated here would represent DE genes between B and CD4 T cells. And this list and this list, they should be highly similar to each other, right? So that's the key idea. Now also, if our candidate annotation wants to say cluster 2 is a novel cell type that does not exist in the reference, then when we do say DE genes for cluster 2, a novel cell type against B cells, and cluster 2, this novel cell type against annotated CD4T, cluster 2 against annotated CD8T, then these DE genes should be very different from the DE gene lists here, okay? Because this is a novel cell type, okay? So now, based on this intuition, so what we do is that for any candidate annotation, okay, we compute DE genes according to the annotation, and then we cross compare with the knowledge we derive from the reference data. So if these DE genes and these DE genes are highly similar, and these DE genes are very different from what we see here, then we'll say that this candidate annotation is likely an accurate annotation. Okay. Now, if we think about how this would compare with what biologists do, okay, so what biologists do when they do manual annotation, they actually only look at so-called cell type specific marker genes, which are derived by differential expression between one cluster against all the other clusters combined, or differential expressed genes of one cell type against all the other cell types combined. Whereas here, what we are doing is we're looking at DE genes between each pair of cell types. So we're looking at more DE genes, right? And when there are a large number of cell types, then there will be many, many lists of DE genes, too much more than what a human brain can handle. So, but we feel that if human beings are, let's say, 100 times clever than our mental state right now, then very likely we can in our mind keep track of all these lists and do this comparison so that we can come up with more accurate cell type annotation. Right? So here, I guess the general design principle is that we want to use DE genes between each pair of cell types and then remember all these but in a way that follows biologists' intuition, if biologists can really keep track of so many DE gene lists in their brain. Okay. And now, once we have this sort of scoring function to evaluate how good a candidate annotation is, and then the next question is, well, we can search the space of all possible annotations and then identify the annotation constellation that optimizes the similarity here and the differences here. And that is the annotation that we want to suggest. Okay, so this optimization is actually fairly difficult. Computer science problem, but we do have a heuristic that can solve this problem. So now, just very quickly, I want to jump to some of the test results we have. So we have, well, in a collaboration with Greg and another faculty member at Emory University, we're collecting single-cell RNA-seq data from ileum samples of Crohn's disease patients, and we're generating single-cell RNA-seq data ourselves. Okay, so this is actually our data shown in a sort of three-dimensional scatter plot, where the three axes shows marker genes for immune cells, epithelial cells, and fibroblasts, and each dot is a cell. So when we cluster ourselves into clusters and then show the clusters along these three axes, we can easily see that each cluster shows a very strong signature for only one of these three tissue compartments. So basically, the good news is, well, with our data, we do clustering, and based on marker genes, we can easily tell which tissue compartment each cell cluster belongs to, and that can form some ground truth for us to test our annotation algorithm. And our annotation algorithm does need a reference data set. So we took a published data set by the Broad Institute. This data set has a fairly large number of cells, and these cells were annotated according to the three tissue compartments, again, epithelial, immune, and fibroblasts. And also these cells are kind of also annotated into 51 more detailed cell types as listed here. Okay, so now we have this reference data set, right, containing X number of cell types, and we have our own data clustered already. So one thing we can do is that we can say take our clusters here, okay, and use our algorithm to map our clusters against the three known tissue compartments, plus also a novel category, because our algorithm has this ability to explicitly label unknown, right? So here is actually our mapping result, and we have a ground truth we can compare to. So our mapping result and the ground truth are actually highly, highly similar, except that for a few clusters that belong to the epithelial or immune compartment, our algorithm incorrectly labeled them as unknown. But maybe there are also some deeper reasons behind that, right? So this is just another version of the results, but well, let me just skip it and show you kind of more impressive annotation results. So again, here, cluster 1 up to 19 are clusters we generated, and then these vertical axes are the 51 cell types in the reference data set. So whenever you see a sort of a spot here, it means that our cluster 13 is annotated as immune plasma cells. So we just copied it here, so that down here we have our annotations of our clusters against these 51 very detailed subtypes. And looking at all these annotations, in terms of whether we get the right tissue compartment correctly, there is actually only one error we made, okay, which is nice. So, but this is basically saying that our algorithm at least can get a correct annotation for major lineages of tissue compartments. But really, the real test is here, okay? So the real test is that, if we take our data and then we perform SARAT-based clustering analysis to identify clusters, and then we use known marker genes to manually annotate the clusters we have, Okay, so this is actually done by a student in Greg's lab. So well, and it's actually a separate analysis of the epithelial compartment only, so that all these clusters are supposed to be epithelial cells. Okay, so in total there are 28 clusters, and here are the manually derived cell type annotation based on no marker genes. Okay, and then what we did was, for all these clusters, we feed them into our automated cluster annotation algorithm against the 51 cell types in the Smiley et al. Reference dataset. And here in this list shows the annotation result from the automated algorithm. And this list here is basically copy paste of the list here, right? So this list down here is the manual annotation result. And when we compare the auto and manual annotation, we notice that among these 28 clusters, 20 of them agree exactly. And 4 of them do not exactly agree, but they are close matches. So for example, progenitor's cell type that's labeled by manual analysis, automated analysis says it's TA2, which is also progenitor cell type, right? So these 4 are close matches, and only the 4 red arrows, okay, these corresponds to really mistakes. So 2 of them, okay, are actually due to insufficient reference, because our reference data set does not have Paneth cells, but the manual analysis labeled these clusters as Paneth cells, right? So then the failure of these 2 of the automated algorithm is actually because the reference data set is not good enough. And the other 2 errors here, I don't have a good way or good excuse to explain them. So those are honest errors that or honest mismatches between the automated algorithm and manual annotation. But then who's to say that manual annotation is correct, right? So whenever there is such disagreement, then we can say that suggest to the biologist who did the manual annotation, maybe you want to revisit this annotation a little bit. Okay, so overall, what we have so far is that we have this sort of algorithmic idea that we feel is sort of doing cell type annotation in a way that's better than the manual analysis, but kind of following the general idea of the manual marker-based analysis that biologists do all the time. And we feel that this kind of extension, right, we do by looking at DE genes across each pair, between each pair of cell types, potentially could capture more knowledge of DE genes or marker genes for the purpose of cell type annotation. And we have shown that in at least this particular example, right, our algorithm is able to achieve an annotation that's highly consistent with what we would have obtained if we're annotating these cell types manually. Although there are a few mismatches, but these there are ways to explain them, right? So one thing that we want to do is that we want to further develop this algorithm so that it will become a convenient tool that people can easily use. And then when biologists are annotating their data, they can maybe run such a tool first to so that they have a set of suggestions in front of them. And then maybe based on the suggestions, they can start their exploration. And that hopefully will help biologists to more efficiently go through their data and more efficiently perform the manual annotation that they may want to do. Okay, so the take-home message here is that, well, automated cell type annotation is, in my opinion, a very useful tool to assist and expedite biological interpretation of single-cell RNA-seq data. And there do exist many automated algorithms that can perform this type of analysis, but we feel there still is room for algorithmic innovation, such as this idea that I just mentioned, combining data-driven and knowledge-driven ideas to perform annotation. I guess for the audience here who are biologists, and who are maybe doing manual annotation in your own analysis, I would encourage you to go through maybe one of the benchmarking papers that were mentioned in the earlier slide, and then maybe among those algorithms, pick the ones that are suggested by the benchmarking, and also ones that are maybe easier to use. Okay, so that you can have some automated way to assist your manual analysis. And for those in the audience who are kind of more interested in statistics and machine learning and algorithmic development, then, well, I think although this topic is a very crowded topic already, I do believe that there is still room for innovation and novel ideas to be explored, especially along the line of how to deal with multiple reference data sets. Okay, so one challenge that I just mentioned is, different data sets as reference, right? They may not be 100%, their clustering analysis and cell type annotation may not be 100% accurate. So that's one challenge. And actually another challenge is that, different, for example, PBMC data sets out there, the annotations that go along with them may have different resolutions, right? So some PBMC data sets are labeled with just the major lineages and major cell types, but then some of those data sets, they are labeled with highly detailed immune subtypes. So then when you have multiple reference data sets about the same biological contexts, but then their annotations do not 100% align with each other in terms of level of resolution, or maybe the same cell type are called different names in two reference data sets. So then in that case, right, how can we consolidate multiple reference data sets in one unified framework that we can use for annotating new data set, right? That is a challenging problem in itself. And that kind of also resonates with kind of one existing challenge about, what really is an atlas. And when we have multiple data sets generated from the same biological context, then how can we efficiently combine them into one comprehensive atlas? One aspect is data integration to remove batch effect, and the other aspect is actually unifying the kind of the initial first pass analysis of these different data sets in terms of their resolution, in terms of their consensus. And maybe these two problems, when both of them are effectively solved, then we'll be able to really come up with comprehensive atlases for different biological contexts by correlating existing data sets. And also, I think another possible direction for cell type annotation is spatial transcriptomics. Well, as we mentioned earlier, spatial transcriptomics data, for example, data generated by 10x VZM, it's not really single cell RNA-seq, it's single spot RNA-seq, and each spot may contain multiple cell types, right? So one question that we talked about was deconvolution, that we want to know for each spot, how many cells are there, and those cells belong to what cell type. In fact, that problem can also be casted as a cell type annotation problem, where we have some reference data set, single cell RNA-seq data set, and then we use that, we can try to learn from it, and then maybe in some way reduce the batch effect so that we can use this whatever model we learn from the reference data set to predict cell type label proportions for each spot in spatial transcriptomic. And so far, I actually haven't seen any paper that kind of try to take cell type annotation algorithms and apply them to solve the deconvolution problem in spatial transcriptomics. So this actually could be an open area that algorithm developers can explore. Okay, so this is my last slide. So let me open up the Q&A window and see whether there are questions. Okay, Oh, looks like the questions were already answered. So let me stop the recording, and well, we can still stick around for Q&A.
Oh, what's the DE method you're using for that, to do all of that? I mean, that's a huge computational burden to compute all pairwise comparisons.
Well, the method I'm using is actually t-test.
Oh, okay.
Which is a sort of not that recommended for single-cell RNA-seq analysis, but it's fast. And also the data that I used, oh, stop recording is here. Okay,